Good afternoon, everybody. Thank you to the organizer to have invited me to give this talk here. It's an honor for me. Actually, I'm a plant pathologist working at the French National Institute of Agricultural Research in France. And I'm working in Bordeaux at the Institut de la Vigne et du Vin, where my research is to actually integrate the evolution and the ecological thinking into the control of grapevine diseases. So in this talk, I will present recent results that we had on a research program on the evolution of grapevine pathogens, especially grapevine downy mildew pathogens, in the context of the deployment of plant resistance. So next. So as everybody knows here, grapevines occupy a very small area of the agricultural area in France and in every European countries, while it needs a lot of fungicide treatments. Here is a mean number in France, like 13 fungicides for a season. And decreasing significantly this number, such as more than half, and the dependency of grapevine upon this fungicide will necessarily involve a breakthrough. And to my opinion, the use of natural plant resistance against pathogen is certainly one of the most promising innovations we have to use in the next decade to achieve this goal. So INRA, like a lot of European institutes, is breeding some new grapevine varieties, very close to the vinifera cultivars we know, with durable resistance to the main pathogens that are powdery and downy mildew. This is for the French program. They combine several sources of resistance, QTLs, and the aim is that they will have good cultural and oenological feature. But of course, we already know that plant resistance genes will not be everlasting, which means that the total resistance will one day break down, and there are also suspicion that partial resistance will erode. That's why we need to have more insight into the durability of these grapevine genes and to understand the potential of evolution of the pathogens. So here is the result we had on the investigating the population genetic structure and the population, the evolutionary potential of grapevine downy mildew in France and in its native area. So we have shown recently that actually grapevine downy mildew is not just one species, but it's a complex of several cryptic species that have specialized on their host. This result has been found while investigating grapevine downy mildew in its native area, which is North America. As you see, there are at least two species that are able to attack Vitis vinifera, Vitis that I called the vinifera 1 and the estivalis. Maybe I should show it. Doesn't work. Yes, it works. Okay. Which means that there are at least two species able to infect our cultivated grape. So we wanted to understand which of these species that have been identified in North America have entered Europe during the propagation of the plant material that occurred during 19th century. So here you see a picture of the genetic variability based on the analysis of one gene for the four species that we have described. Okay. And in white, now I will show you the diversities that have been observed in Europe, contrasted to the whole diversity of the cryptic complex, which is called Plasmopara viticola at that time. So we have entered very little variability of the pathogen in Europe. In Europe, the genetic structure is very low. We had only two clusters, the red and the blue one in the slide. low but significant differentiation, probably that result from a leapfrogging event from Western to Eastern Europe during the last century. Also, an important result is that we found that the pathogen is able to achieve very efficient sexual reproduction, but this is already known from oosporic observation. And finally, we found that the pathogen, Europe actually, serve as, let's say, a bridgehead center to disperse the pathogen, the colonists from Europe to South America, South Africa, Australia, and even China. So, what about the plant resistance now? Actually, we can say that there is a risk that a reintroduction of the pathogen into Europe will complexify the situation. Hopefully, it will not happen. Still, we have to be careful. Second, we have found that the pathogen is able to undergo sexual reproduction in here, which may provide him a lot of potentiality to adapt to new conditions. Third, well, there are large effective population size, which are due to this mixed system of reproduction, and we can conclude that the pathogen has a high evolutionary potential. Still, what do we know about its adaptation to the host plant resistance that is going to be deployed in Europe? To explore this question, we have actually an approach which is based on the sampling of the pathogen, cross-inoculation experiment in laboratory condition, plus quantification of life history traits of the pathogen. So what do we do? We have sampled, for instance, in this experiment, some isolates on resistance variety, here the res isolates, and on vinifera cultivar for the sus population in different locations in France, Germany, Switzerland, Ticino, etc. And we have cross-inoculated the strains and the plant, choosing three resistance varieties which are well known from the Germany breeding program, Bronner, Prieur, Regent, and control, which is the Cabernet Sauvignon. So this resulted in a huge unique bioassay with 103 isolates that have been crossed on 4 different host plants, and we measured sporangial production, sporangial size, etc., at that time period. I will not enter the details. Here is the main result of the experiment. So as you can see, so I could show it. This is the result on Cabernet Sauvignon, which is our control, a sensitive host. So we observe this reduction of sporulation of the pathogen when inoculated of the resistance variety. Here are the name of the resistant variety, Regent, Prieur, and Bronner. But the second result is that population in dark green here that were collected on the resistant varieties, so that had time to adapt, were more aggressive on these resistant variety than the naive population collected on Vitis vinifera that have never seen the resistance genes, which means that the pathogen is slowly adapting to this deployment of resistance. This is obvious, and this has happened in very short time. It means less than 5 years. The result is not due to the spread of one specific genotype that could be like a clonal expansion of a very adapted lineage of the pathogen. Instead, we found no genetic differentiation between the isolates that have been collected on resistant and susceptible hosts, as you see in these graphics. No genetic marker to disentangle between these two populations. And the last result, which is maybe the more important for me, is that this adaptation does not seem to be specific to the variety on which the pathogen has been collected. It's more like a general adaptation to resistance, resistance in this variety is partial, it's quantitative, and the erosion of resistance seems to select for pathogens with higher aggressiveness that are able to be more fit on every resistant variety, all the resistant variety. There is no specific adaptation to the variety of origin of the pathogen. Well, this is just to summarize the results. So we have isolates that have been selected here on the resistance, have shorter latent periods. I didn't show that result. They sporulate more, and their sporangia are smaller than the naive isolates that we already know on Vitis vinifera. Moreover, there are no fitness costs associated with this greater aggressiveness, which means that they are higher, they are more aggressive, and there is no cost for it. And they do not adapt to their variety of origin. However, we can still use this resistance to achieve a different cropping system, and as I said, it's probably one of the most promising idea and innovation that we can integrate in the cropping system. But the plant resistance will not solve all the problems we have to control the pathogen. It's only a new component that is added to the system, which is likely to modify in depth the winemaking process. So here is a slide just to show this, to illustrate this idea. So we are starting from this system where we have chemical control, biological and plant manipulation, and we will move to a more complex system and a more balanced system where plant resistor is integrated, with the aim to, of course, reduce the chemical control. So plant resistance will have to be complement, because for a lot of pathogens such as downy mildew, there is no complete resistance. So this efficacy has to be completed in order to secure the production, whatever the epidemic's condition. We will have also to control the pathogens that are not targeted by the resistance. And these diseases are increasing because the reduction of fungicide, which is subsequent to the use of plant resistance, will favorize diseases like black rot, for instance. And also we have to keep in mind that we have always to limit the adaptation of the pathogen populations, such as, for instance, limiting their population size. Here is an example of disease epidemics that you can observe when you do not treat a resistant variety. Here it's a French resistant variety, total resistance to powdery mildew, partial resistance to downy, and you see an increase of back rot on clusters immediately after 3 years. It's a complete, complete destruction of the yield, and also an increase of downy mildew on leaves after 3 years of absence of treatments, which means that we have to develop strategies that control black rot and downy mildew, especially at the end of the season. So we are testing some decision rule systems. I will not enter the detail of this program, which is in itself would deserve a complete presentation. But for instance, we have compared the 3 strategies, a systemic strategies, a curative one, and a preventive one, let's say, to control the 2 pathogens, black rot and downy mildew, on this French resistant variety. And here are the results. First result is that there is no yield if you do not control the pathogen with fungicide due to black rot epidemics. This is clear from the control that you see here. While the 3 strategies work quite well, with differences between them in the number of treatments, between 2 or 3, depending on the strategy, and different results also in terms of severity and incidence, especially for black rot on cluster. And finally, what do we do? We have actually launched a new long-term system comparison where we assess agronomical and economical performance of these varieties. This is a system that has been planted in 2011 in Bordeaux with replicates of big size, 0.2 hectares for each replicate, where we assess yield, disease prevalence, labor time cost, etc., to have an idea of what's going to happen. And just a quick result on this long-term agronomical cropping system comparison to show that there are a lot of hope with these new varieties, because as you will see here, compared to a reference for France and Gironde, especially here, and to organic system, classical one, not very high-tech one, the resistance system we had launched here is able to reduce by 80% the number of treatment with a good yield at the end of the season. That's it.
Just a moment. Se c'è giusto una domanda adesso, così prima di cambiare relatore.
Tutto chiaro?
Tutto piano.
Neanche io non ho sentito.
Scusi, ci sono anche varietà come Cabernet Sauvignon o Merlot che sono resistenti? Per adesso soltanto Regento.
Cabernet Sauvignon and Merlot are not resistant.
No.
Okay. No.
Alors il n'y a pas encore de sorte Merlot et Cabernet Sauvignon qui sont résistants.
No.
On n'est pas encore arrivé à ça.
And they will, if something, if the new variety is going to be very close to Cabernet Sauvignon, they will not give this name actually.
So the wine taste is completely different. You cannot make the
this
same
is
wine,
one of the
so.
questions I do not enter here.
Okay.
But of course, it's a very important agronomical performance and enological performance. But the idea is to breed to have very varieties that are not interspecific hybrids. They are back-crossed several times in order to be very close to the vinifera type, while keeping the resistant genes from the vitis, Asiatic vitis or American vitis, depending on the cross.
Scusi, siccome qui parliamo di innovazione, allora la domanda sorge spontanea. Lei dice la resistenza non è per sempre, addirittura è in grado di isolare cepi di patogeni.
Translation.
Ok. Merci beaucoup. Se può ripetere la...
Sì, è una domanda in tema con l'oggetto di questa serie di conferenze.
Channel 2. Okay. Channel 2, right? Yes. Sorry.
Prego. Posso parlare? Grazie. No, siccome qui parliamo di innovazione, quindi di strategia, se le varietà resistenti a lungo andare, a breve per la verità, non tanto a lungo, non mantengono la resistenza, anzi aumentano la virulenza dei patogeni, la mia domanda è: che senso ha introdurre questo tipo di innovazione nell'attuale contesto? Perché o diventa un senso sistemico e allora dobbiamo cambiare tutta la viticoltura sulla base di queste varietà, ma lei ha dimostrato che è assurdo. Oppure tanto vale studiare forme di resistenza interne alle nostre popolazioni e non portarci a una modifica di un modello che di per sé stesso non ha durata e rischia di compromettere il modello attuale, in termini commerciali intendo.
Okay. Actually, I will not. I can only give my opinion on that and not answer the question because it's a full discussion that you have open. But of course, my results are probably a bit too much pessimistic about these varieties. Also, you have to, because the pathogen, of course, the pathogen is adapting, but it's also adapting to fungicides, as you know, and you still can manage it with fungicide also while there are resistance in populations. So it's more or less the same debate here, and I think that provided, given the last slide I've shown here, that it's worth trying to innovate on this in this line if we want to move towards a real decrease of fungicide use. It's my opinion, at least. And last point is, my talk is more to warn about the risk of using them, but we know that resistance that have been eroded is still efficient in the field, especially when the epidemics is not too high. And if we include these plant resistance and combine them with fungicide, I'm not saying that you will never treat for pathogens of grape, but then you can achieve a new model, a new cropping system that is really innovative from my point of view.